Welcome to this tutorial on DNA extraction, using the MagBeads FastDNA Kit for microbiome applications, compatible with the MagFlex-96 automated nucleic acid purification system. The MagBeads FastDNA Kit for microbiome can process a wide range of sample types, including microbial cultures, bodily fluids, and environmental samples. In this demonstration, we'll focus on swab sample extraction using the MagFlex-96 for automated DNA purification. Let's begin with sample preparation. Add the swab sample to a lysing matrix E tube. Then add 815 microliters of buffer MGB1 and gently invert the tube several times to mix. Homogenize the mixture twice, using a fast prep instrument, 5 meters per second for 45 seconds each, with a 5-minute interval between runs. After homogenization, centrifuge for 2 minutes at 15,000 G. For samples that lyse easily, such as biodiversity swabs, a single 45-second lysis at 5 meters per second is sufficient. Transfer the supernatant to a 2 milliliter microcentrifuge tube. Add 300 microliters of buffer MGB2. Invert 5 times and centrifuge again for 2 minutes at 15,000 G. Next, we'll perform automated DNA extraction using the MagFlex-96 aNAP system. Add 400 microliters of the lysis mixture to each MagFlex plate well. For example, positions 1 and 7, or 3 and 9. Place the plate into the MagFlex-96 instrument. Insert the MagFlex rod sleeves into well position 2, or 8. Run the preset program, DNA Microbiome, or manually configure the instrument using the parameters shown. The MagFlex-96 will automatically perform bead loading, DNA binding, and washing at room temperature, followed by a heated elution step. Once the program is complete, transfer the clear eluent from well position 6 or 12 into a clean 1.5 milliliter microcentrifuge tube. The extracted DNA is now ready for downstream microbiome analysis, or can be stored at -20 degrees Celsius for future use. The MagFlex-96 streamlines DNA extraction through precise magnetic bead handling and automated processing, minimizing hands-on time and variability. Its high-throughput 96 well format ensures consistent results for microbiome studies that demand both speed and reproducibility. By integrating the MagBeads nucleic acid extraction kits with MagFlex-96 automation, you achieve reliable, high-quality DNA ready for applications such as sequencing and metagenomic analysis. To learn more, visit www.mpbio.com.